invite everyone to today's talk. And I'll be introducing the first speaker of our of the first talk of today. The first speaker is called Elvis Temfack. Is from Cameroon, Yaoundé, I think, according to the information I got. So Elvis is a medical doctor at Douala General Hospital, Douala, Cameroon. And he also has another variation at the National Public Health Laboratory in Yaoundé. He's a PhD holder in infectious diseases and microbiology from Paris Descartes University and the molecular mycology unit of Pasteur Institute. He has different areas of expertise, among which include comprehensive patient care, clinical research, and epidemiology. He's an active member of the HIV and AIDS Therapeutic Committee of the Douala General Hospital. His main interests are in molecular mycology, clinical research, and microbiology. Today, Elvis will be talking to us about HIV-associated Cryptococcal Meningitis, a pragmatic approach for resource-limited settings. I invite you, Elvis, now to give us this beautiful talk. Thank you.
Okay, thank you very much, everyone, and thanks to the organizing committee for inviting me to have this exchange with the medical mycology trainees. So my talk is going to be, I'll try to do an overview and the objectives of the talk. Oh, sorry, my slides are just a little bit big. So the objectives of the talk would be mostly to understand the burden as well as the preventive as well as diagnostic methods that we have. Then we'll do an overview on recommended options of treatment. So just a quick reminder, Cryptococcus is a free-living yeast that we can find in avian feces as well as trees, barks of trees like eucalyptus in nature. And there are two main species, Cryptococcus neoformans and Cryptococcus gattii. So initial infection is through inhalation of the desiccated yeast into the lungs. And when they get into the lungs, either they can be resolved due to a potent immune system, or they may be phagocytized and stay latent in macrophages and wait until there is some sort of cell-mediated immune depression, where they become secondary disseminated. And when they get to the brain, it causes one of the most dreaded and one of the most fatal clinical presentations of the disease, which is cryptococcal meningitis. So looking at the risk factors of cryptococcosis, as I mentioned earlier, it's mostly found in people with profound impaired cell-mediated immunity. So looking at our discussion of today, we have two groups of people: non-HIV related cryptococcosis that we'll find mostly in solid organ transplant patients, patients on immunosuppressive therapy, as well as those with hematologic neoplasia. So just to share with you some data about HIV-related cryptococcosis, when we look at some surveillance data of the National Center for Invasive Mycosis in Institut Pasteur, we see that at the end of the 80s and the beginning of the 90s, there was really a surge in the total number of cases of cryptococcosis. But when we look at the numbers associated with HIV, this increase was first associated to immune depression with HIV. However, in the early 90s, with the introduction and the scale-up of antiretroviral therapy, the total number of cases decreased, and there was more or less a plateau that was similar to the numbers before the 80s. And in the patients with HIV-associated cryptococcal meningitis, about 50% were found in HIV patients. However, in Africa, more than 90% of cryptococcosis is found in patients with HIV. And in these patients, the median CD4 is around 45 cells per milliliter. And this is the threshold at which we say the patient has advanced HIV. So what is the burden of advanced HIV, which is the major risk factor for cryptococcal meningitis in Africa? We all know that following HIV infection, if no treatment is given, there is natural progression to advanced HIV. And when the CD4 is less than 200, that is a threshold where many opportunistic infections occur. So if we look at UNIAIDS data of 2020, we find that among the 38 million people who were living with HIV, about 12.6% are at risk of advanced HIV because they are not yet on treatment. And even those who present to care, 30 to 40% of ART initiators already have advanced HIV. And in this group, it's already clear in WHO guidelines that they should be systematically screened for cryptococcal meningitis prior to introducing antiretroviral therapy. On the other hand, among the 25 million people accessing ART, we see that there's a high proportion of them who report to health facilities with advanced HIV. And this up to about 4 out of 5 of them in hospitalized patients with HIV on antiretroviral therapy are presenting with advanced HIV, which brings us to the situation where we have to say there is need to actively screen all unwell HIV patients who are on ART in search of advanced HIV, and given our current discussion, looking for cryptococcal meningitis. When we look at the burden of HIV-associated cryptococcal meningitis, it was estimated at more than 223,000 in 2014, among whom about 181,000 died. And in this, among those with cryptococcal meningitis or those who died, 3 out of 4 were found in Africa. And when we revisit the causes of mortality in HIV patients at the end of 2019, we see that the first cause was due to tuberculosis, and next on the line is cryptococcal meningitis. And cryptococcal meningitis represent about 15% of all HIV-related mortality. And in some settings, mortality goes up to about 80% of those who have the disease. And currently available evidence shows that pooled mortality in routine settings is about 44%, which is about 1 out of 2 patients, which in 2021 is unacceptably high. So how do we diagnose cryptococcal meningitis? So I would talk about proving cryptococcal meningitis the diagnosis is only made when there is phenotypic identification of yeast cells in cerebrospinal fluid collected through lumbar puncture under microscopy after Indian ink staining or from culture media by microscopy. And when observed, we see a rounded cell with a halo around the cell on an Indian ink background. This halo represents the cell wall. And if we look at the particularity of this cell wall as compared to other interesting human mycosis, we see that Cryptococcosis has a very thick cell wall, which is very specific, and this cell wall contains complex of polysaccharides, which can be detected in body fluids if Cryptococcosis is present. So this specificity of the cryptococcal antigen is something which is quite important in the diagnosis as well as the follow-up of the disease. How do we detect cryptococcal antigen in body fluids? So in the early 60s, the first approach was to use latex agglutination, which required some laboratory storage, some laboratory experience, and it was really very subjective because it needed some agglutination. Then came up objective ELISA, but this requires a lot of technological know-how as well as continuous energy to be able to do the test, and it's very difficult to do on routine settings on 1 or 2 samples. You need to collect a batch of samples to do them. And then of recent, there was the introduction of the lateral flow assay, of which currently there are 2 which have been currently commercialized. The particularity of this test is that it needs minimal training, can be done by a nurse. The results are obtained in 10 minutes. It can be stored at room temperature, and the shelf life is very high. So this is the type of test that we can find in really very remote areas. And the interesting thing about cryptococcal antigen is that it can be detected weeks to months before the onset of the disease. And this particularity is something which can really be exploited for the pragmatic approach in the management of the disease. If we go back to the rapid advice provided by WHO in 2011 on the prevention of cryptococcal meningitis. At that time, it was really clear that routine and primary antifungal prophylaxis should be avoided in patients with less than 100 CD4 count. And it was suggested that every patient with less than 100 CD4 count should undergo serum cryptococcal antigen screening, and those who are positive with no evidence of cryptococcal disease should be put on preemptive fluconazole therapy. So this has been practiced in so many countries in Africa, especially East and South Africa. And so we were interested to revisit this recommendation to see whether there's an impact of this on the incidence of cryptococcal meningitis. So we were comparing incidence of cryptococcal meningitis among patients who were CAC positive as well as those who were CAC negative. And interestingly, it was found that without any preemptive therapy, patients who were cryptococcal antigen positive had a risk of about 21% to develop cryptococcal meningitis within the first year of antiretroviral therapy. And among those who received some prophylaxis, irrespective of the dose or the duration, this is not shown on this slide, this risk dropped to about 9%. So among those in whom a lumbar puncture was done to exclude underlying cryptococcal meningitis before preemptive therapy, the risk of developing cryptococcal meningitis within the first year was equal among those who were CAC positive and CAC negative. And then, so among those where lumbar puncture was done, 1 out of 3, though asymptomatic, was found of having underlying CSF evidence of cryptococcal meningitis. This made us to conclude that underlying undiagnosed asymptomatic cryptococcal meningitis explains the excess risk of cryptococcal meningitis in CAC positive patients. We also looked at mortality, and we know very well that in patients with advanced HIV, mortality is very high. And in these patients, it was about 3 times higher among those who were CAC positive. And even with lumbar puncture to exclude cryptococcal meningitis and preemptive therapy, mortality risk was about 2.2 times higher among those who were CAC positive as compared to those who were CAC negative. Given that this is a discussion with mycologists, I think this gives us a question: why is it that these patients who are CAC positive are more likely to die than those who are CAC negative? Is there any interaction between Crag and immune response, even on antiretroviral therapy? That is a good question, which could be used as a research question. So when we look at what screening of cryptococcal antigen can bring in our pragmatic approach in diagnosing the disease, we see that using serum Crag screening among Crag positive patients, as I already mentioned, 1 out of 3 already had evidence of cryptococcal meningitis in CSF. And the risk of developing symptomatic disease is about 21% within the first year of antiretroviral therapy. And we also know that symptoms precede serum Crag positivity by a median of about 22 days. And among these symptoms, we have things like headache, confusion, altered mental status, which means that among symptomatic patients, if we do a Crag screening, which becomes positive, and then we do a lumbar puncture, we are able to diagnose about 40% of those with cryptococcal meningitis, as we described in one of our recently published meta-analysis. This tells us that serum Crag is a surrogate for timely diagnosis of cryptococcal meningitis and can permit us to diagnose up to more than 70% of the cases, even in the most remote areas in Africa. So with this, we suggest as a pragmatic approach that any patient who presents to the hospital with symptoms of which may be suggestive of cryptococcal meningitis, we start with a serum Crag, and if it's negative, we can think about other causes of the observed symptoms. And in case of positivity, we're in a setting where we can do a lumbar puncture, because in many cases, a lumbar puncture, and then we can be able to do laboratory analysis. If the test is negative, it brings us to the first option of considering other causes of the symptoms. But in case the test is positive, we can go ahead and induce combination antifungal therapy, which will permit us to institute the treatment as early as possible. But we know that in some settings, lumbar punctures are not possible because the patients refuse. There may be a lack of material, or it may be strictly contraindicated. In this case, if the patient is CAC positive, considering to put the patient on antifungal therapy, combination antifungal therapy, especially if this patient was previously known to be CAC negative, it's not a wrong approach. And in settings where we can do Indian ink and staining, if we do a CSF crack testing and it's positive, we induce treatment. And if it's negative, we consider other causes of the symptoms. So now that we've looked at how to diagnose the disease, how can we treat? So in 2018, WHO updated the guidelines for the treatment of cryptococcal meningitis based on the results of the ACTA trial. And this suggests that we, as always known, the disease will be treated into 3 phases: the induction, the consolidation, and the maintenance phase. However, the particularity is that in the induction phase, instead of 2 weeks amphotericin B, it's 1 week plus flucytosine, followed by 1 week of high-dose fluconazole, or the option of using oral combination therapy, flucytosine and fluconazole, which is very easy to administer in cases where especially in remote areas where hospitalization and the use of amphotericin B could be difficult. The consolidation phase is still with high-dose fluconazole, as well as the maintenance phase with 200 milligrams of fluconazole till the patient has more than 200 CD4 counts. We also have to know that when we see the culture of crypto in the lab, the same jelly type, jelly type appearance that we have in the petri dish is the same that may be found around the meninges. We know that cerebrospinal fluid is produced in the choroid plexus inside the ventricles, and it's resorbed in the villi around the meninges. So in cases of cryptococcal meningitis, there can be an increase in the intracranial pressure due to poor resorption of CSF, and this can happen at presentation, or it can happen in the course of the treatment because of massive killing of the yeast. And in this case, symptoms have to be monitored and managed, as we recently showed in a paper published at the end of last week, that high fungal burden as well as raised intracranial pressure are independent associated with visual loss in HIV-associated meningitis. And these are factors that lead to high mortality. So the management of raised intracranial pressure relies on therapeutic lumbar punctures. I will repeat, and I will never say this enough, therapeutic lumbar puncture is the way forward for the management of raised intracranial pressure. And the objective is to relieve symptoms, to bring down the pressure to normal, or at least 50% of the initial pressure. And this should be done every single day when the patient complains of symptoms, among which headache, or when this patient is unconscious, or is confused, or has visual or hearing impairment. And we can remove up to 30 milliliters of CSF. And in cases of cryptococcal meningitis, please avoid steroids. Diuretics would not work for raised intracranial pressure. So there's always this question about when to introduce or to reintroduce antiretroviral therapy in a patient with cryptococcal meningitis. We acknowledge the fact that most of these patients with advanced HIV, some of them have TB, and TB recommendations say the lower the CD4, the earlier the start of antiretroviral therapy. But for cryptococcal meningitis, we need to defer antiretroviral therapy by 4 to 6 weeks after we've initiated antifungal treatment, because of the high risk of immune reconstitution inflammatory syndrome. We elaborated this very clearly in one of our recent papers that we shared last year, 2 years ago. So given that I mentioned the fact that cryptococcal meningitis is the second cause of mortality among HIV patients, this discussion is among mycologists. I just want to draw your attention to the fact that the future of curbing mortality associated with AIDS is our direct responsibility as mycologists. Why do I say so? There is growing evidence that the burden of histoplasmosis is comparable to that of TB. And now in the Amazon, colleagues are asking the question, histoplasmosis or tuberculosis in HIV infectious patients, which one should we treat first? This tells us that maybe part of the excess mortality reported for TB is due to undiagnosed and untreated histoplasmosis. I just want to challenge all of us that when we look at the reports about mortality associated with HIV, there is nowhere where histoplasmosis appears. This doesn't mean it doesn't exist. So I'm bringing back this discussion to us that we should consider looking for histoplasmosis in cases of suspect of TB treatment, of TB infection. So at this juncture, I will just summarize by saying that HIV-associated Cryptococcal meningitis is highly fatal if not treated, and current availability of point-of-care tests makes diagnosis relatively easier, and availability and adequate administration of currently recommended treatment is a cornerstone of survival. Monitoring and treatment of raised intracranial pressure improves survival massively. I will end by saying there's a current reported high mortality associated with HIV-related TB, masking undiagnosed and untreated histoplasmosis. Please permit me to acknowledge quite a few people from these institutions who contributed to the work I've just shared with you now. Thank you very much for your attention.
Thank you, Elvis. Thank you, the audience, listening in of the two talks. So I'll pass you to my colleague to introduce the second speaker of the day.
Hi. All right. So, hi, my name is
You,
Rebecca. Oh, thanks. Sorry. I was hearing you a little bit choppy. I'm not sure if that's my connection. So hi, I am very excited to welcome our next speaker today. Our next speaker is Hungji Tsai, who is originally from Taiwan, received his bachelor's in agricultural chemistry at the National Taiwan University, then completed his PhD at the University of Minnesota, where he worked in Dr. Judy Berman's lab on centromere and origin organization in Candida albicans. So while he didn't study aneuploidy or drug resistance in Candida while in Judy's lab, he liked the biology enough to then join the lab of Rong Li at Johns Hopkins University, where he worked on aneuploidy. So although the lab focused on budding yeast and cancer cells, he then started chemical screening to target aneuploid Candida albicans. And his postdoc eventually led him to think about how biophysical cues affect the evolution of drug resistance. So as the main focus of his research group. So in mid-2019, after the end of this postdoc, he visited an immunology lab, Dr. Larman's lab at Johns Hopkins University, to develop new genomic tools for antigen discovery. And very recently, as of 2020, Dr. Tsai started his own group in the Institute of Microbiology and Infection at the University of Birmingham in the UK. So their very new research group studies mainly fungal pathogens with ongoing genome and chromosome instability and how this is involved in survival of antifungal stress and interactions with the environment. I will add as a plug that Dr. Tsai's brand new lab currently remains empty due to the pandemic, but will be actively recruiting students and a research tech position in the future. So keep an eye open for that. And I'm very happy to welcome Dr. Tsai. Take it away.
Okay, can you see my screen well?
Perfect.
Okay, thank you, Rebecca's nice introduction. So I'm Hongji Tsai, and first I want to thanks to the organizer to give me this opportunity sharing the story I have been working on and what I am going to working on. So no, I should say we are going to now. So today we are going to talk about genome instability and genome plasticity. This is very vague to me between these two terms. By the end of this talk, you might understand why I feel like this is a very vague term. It's lucky that Elvis gave a very nice talk and address the clinical importance of fungal infections. And so for this audience, I don't need to address that again. So let's just go into straight the point, that genome instability and how it plays as a major role during the evolution of antifungal resistance. So since we only have 3 antifungal drugs in the past 30 years, so antifungal resistance is kind of inevitable problems we are facing. But why this is so difficult to treat? One of the reasons is that we actually really don't know that how this happens. We all know that there are adaptive mutations. We all know that there are a lot of different stress in the host or antifungal, which can trigger genome instability, fungal genome instability, and to push them to evolve to become resistant, since mutations, genome instability is a major force to drive evolution. But how this happen and how a mutation, think about like a mutation mostly cause the cell with a detrimental effect, okay? It has a fitness trade-off. But how come that a mutation or a large mutation can give them advantages, okay, to survive under antifungal drug? So in the past few years, we are studying aneuploidy. So this is one of the major cause to cause antifungal resistance, since it actually quite common to be observed in a lab or wild yeast or a lot of clinical fungal isolates. But aneuploidy is most well known from the general population, like Down syndrome, which cause really severe fitness defect at the organism level and also cellular level, of course. And this kind of phenomenon that have gain or loss of chromosomes, how it eventually become a major cause of the antifungal resistance is just not really known. So today we kind of feel like we finally understand a little bit. So let's start from why aneuploidy is a driver for drug resistance. Okay, so as I mentioned, that aneuploidy is again a genome type with gain or loss of chromosomes. That means that this can be happen in one cell cycle, one cell division, and this process can be very random. Okay, so I mean, sorry, the consequence could be very random, because if like for example, in Saccharomyces cerevisiae, there are 16 chromosomes. So a missegregation event can cause 1 million different possibilities in this case. And from the study in 2010 from Norman Pabalka, he created a panel of aneuploidy, stable aneuploidy karyotype, and bring them into and bring stress into them. Okay, they grow the aneuploidy and euploid into different kind of stress. Okay, this wide range of the stress mostly cause quite significant fitness defect to the euploid cells. But when you compare their fitness difference, almost every stress, okay, when the cells grow in every stress, there are always 1 or 2 aneuploidy isolate strands can grow better than the euploids. Okay, so that means that aneuploidy actually drive this kind of random event actually drive a large phenotypic variation, and it kind of invincible, okay, to adapt to stress. So this could be so how it linked to drug resistance. So in 2006, Anna Somaki in Judy Burnham's lab collect a large collection of Candida isolates from the clinical patients. And they found that when those clinical isolates doesn't have any aneuploidy chromosomes, it's still quite sensitive, okay, to the fluconazole. And this is a fluconazole strip from high concentration to low concentration. But this is a situation microarray. But if you can see a copy number gain on the left arm of the chromosome 5, this clinical isolate actually can grow on the fluconazole treatment regardless of the concentration. And when they take this strain back to the lab environment and grow them in the optimal condition, so this chromosome can be lost. and then become sensitive again. This provides the first direct evidence that aneuploidy can drive drug resistance. And later, when Roman Pavaka and Guangbo Chen in Rongli's lab, when they collect this big the big collection of aneuploidy strains, they also can see that this aneuploid population can grow way better than the euploids under different stress conditions. So these become really troublesome because if aneuploidy can create a random karyotype, really random genome type, and happen in one cell division, does that mean that antifungal drug, if this happen in the host, an antifungal drug cannot really treat the fungi anymore? So to stop antifungal resistance, the big question is, how can we target aneuploidy? Okay, how can we target these random evolving populations? So study aneuploidy is just like you are collecting a Pokemon, okay? This is from the Pokemon Wikipedia. So this is, I feel this is a very good analog because each Pokemon has their own ability and they look very different. And although if they are in the same kind of catalog, they look similar, but they still have different kind of morphology. And if you give them some stress, they may change again. So this gives us three challenges when we study aneuploidy. One is that aneuploidy genome state actually is already highly unstable. So they can keep changing, okay? And each individual will generate a different phenotype because they have different genome type. So they drive karyotype specific phenotype. And in this case, when you try to study aneuploidy, okay, how many strains you need to collect to find a common signature? 10, 100, or 500? So we don't know. So, but at the beginning, we think, okay, we can leverage this. We can leverage this challenge that there is a karyotype specific phenotype. So we all know that Fucazo can, the cell Candida can with isochromosome again can survive under Fucazo. That means that we can culture the Candida in Fucazo and have the entire heterogeneous population become into only cells with chromosome 5. Then we can find a drug, okay, to target this specific strain. So we do a chemical screening years ago and call this evolutionary trap. And we indeed can find a compound among all 4,000 different FDA approved compounds. Like this compound, when they treat with the euploid, the diploid Candida, there is no fitness change, but it will kill the Candida with isochromosome 5. But think about this. This is a proof of principle concept, but there are hundreds of genes on the chromosome 5. What these drugs target, we don't know. We have no idea. So if we go back to think about the mechanism, we still don't know. So that's the time I really switch back to the Saccharomyces cerevisiae because it has so many tools I can use and try to search what's the common biological signature. So the tool I can use majorly is because saying like we can leverage the biotic process to generate aneuploidy progenies. So when there is a 3N, okay, triploid mother, when I induce the meiosis, in the meiosis one, there will be a random segregation happen. So it will lead to aneuploidy progenies. So in this case, I simply just throw the cells into the sporation medium and wait for a couple of days. Then I can dissect thousands of the aneuploidy progeny to study. So long story short, we grow this culture, okay, we grow each individual aneuploidy progenies individually in a plate, and we pull them together and try to see what is the common signature. Because we believe that when the population is heterogeneous enough, a lot of phenotype can be canceled out regarding the transcriptional profile. And if there is a common signature, okay, it will keep add up, okay? Eventually we can detect it when the size is big enough, regardless weak or strong in the individual strains. So luckily, 20 years ago, Ojigashi actually used more than 100 different stress condition to treat the yeast cells and do a lot of microarrays to understand what is the transcriptional response, okay, to different kind of stress. And so we try to search what is our aneuploidy transcriptome look like, okay, which stress has the highest similarity, okay. So we found that hypo-osmotic stress is the one, okay. Aneuploidy cells seems display a transcriptional profile similar to cells under hypo-osmotic stress. So what is hypo-osmotic stress? It often happens that when there is a osmotic imbalance and there is a huge water inflow into the cells. So it will cause an increase of turgor pressure, and then it push the cell surface from inside, okay, because the water just keep going in. And then it will cause the cell wall or membrane stress. And eventually the cell will swell. You can just imagine like a overblown balloon, okay? So the first thing we test is we use AFM, the atomic force microscope, to look at the cell surface stiffness, because if there is a force to push the cell surface, okay, so the cell will become stiff, okay. So indeed, aneuploidy do have higher cell surface stiffness, and if that's the case, if we digest the cell wall, okay, this turgor will push the membrane, and the cell will burst way faster than the haploids or other euploids, and this is also the case we observe. So, but what does that matter, and why it changed? So what is change? This cell surface, why it changed so much regarding the cell physiology? So one of the most dynamic organelle is plasma membrane. Okay, it sends inside and outside. Okay, so it keeps changing. So, but if there is a physical force push the membrane, that means that the proteins or membrane-wise, they cannot endocytose very well, internalize very well. So we stand with the cell membrane, the lipidi, and try to observe their internalizations. And we indeed can see there is a delay or completely compromised regarding the membrane internalization. But this could be because of so many things, right? And and if this is indeed an osmotic imbalance, we're supposed to be able to rescue it by adding more osmolytes in the environment, in the media. So we add sorbitol, and we can see these. We can see this internalization process, okay, being rescued. So we found this is extremely interesting because what we are talking about is a nuclear genome instability, okay? It generates a biophysical force, okay, to change the membrane structure, to change the cell homeostasis. So, but where is that from, right? So one of the known common signature of aneuploidy is proteome imbalance, because not every chromosome increase their copy number, okay? So when there are so many protein complex inside the cell, there will be a lot of protein subunit being made, extra protein subunit being made from the aneuploid chromosome. And those subunits cannot go anywhere. They don't have partners to bind each other. So they're floating around. So it's just like what we see in the textbook when we went to the high school, that when you have two chambers with a semi-permeable membrane, you have more osmolyte here, you have more solute here. Water will influx into the other side to balance. So we take the proteome, we go to the proteome database and try to look at the protein complex number, the subunit number in the protein complex, and try to simulate these results and try to look at the cell size change versus the ploidy level change. So you can see that in the euploid 1N, 2N, we actually have 3N, 4N here. It's a linear change, but the cell size actually change in a non-linear format manner when they are become aneuploids. Okay, so and this is consistent with what we saw in the experimental results. And this kind of phenotype also can be observed in other eukaryotic, also from some database searching. So to give you a little bit summary, so we see that aneuploidy, a nuclear genome instability event, actually it lead to a hypo-osmotic stress state. and generate the turgor pressure to cause cell surface stress. And this kind of plasma membrane, this kind of biophysical force actually change the plasma membrane turnover. It change the transporters or lipid, all different things, compositions on the plasma membrane. And this actually is a predictable biophysical outcome, okay, when the aneuploidy happened. So now I actually trying from those story in my postdoc. So we indeed now knowing that, okay, that's what happened when the genome instability happened, and how it caused the cellular physiology change in the fungal cells. So I give that a check. But there are so many steps here. How this biophysical force change, and how does the aneuploid, so how the aneuploidy play a role during the evolution of drug resistance. So this become the major topic of my future research program. How this large-scale genome instability contribute to the genome plasticity. Yeah, we don't know that. But one of the phenomena we see, and actually is one of the reasons we start going to this direction, is that when we do the generate aneuploidy progenies, these aneuploidy populations, we actually can, because this is yeast, we actually can monitor their growth, and we actually can consider if we can add a single gene mutation in each allele, okay? So we can generate a heterogeneous aneuploidy population, carry the same single gene deletions. So in this case, we have deletion collections. So I can keep doing this, and we collect like 5,000 different genes, okay? So in each whale, there are 50 plates. In each plate has 96 whale, and each whale has an aneuploidy population. And each population carry the same, has a single gene deletion. So in this case, I can actually study, okay, is any gene mutations actually promote aneuploidy, okay? Or is any genes can any genes cause the diminish of aneuploidy cells? So we look at these growth, and we compare the fitness. We generate a growth curve, compare the fitness ratio between aneuploidy population and the haploid population, okay? Carry the same gene. And we saw this. This is interesting because when we compare aneuploidy versus euploid, we expect that aneuploidy grow worse, or there could be one or two genes, okay, grow better, cause aneuploidy grow better. But in fact, 60% of the genes, when it got deleted in the aneuploid population, they grow better than the haploid with these single genes. So how these fitness laws got rescued, we really don't know what happened. And in 2005, there is a really nice study which in Candida albicans isolates, from clinical isolates. This study, they collect the clinical isolate from the single patient and in different time, and try to see how Candida evolve resistance. So they found that aneuploidy can be transient, okay, because of the high fitness trade-off. So but the conclusion is that the transient aneuploidies may all confer a more stable, okay, adaptive phenotypes. So that actually fit that there can be a lot of genes when they happen, they occur in the aneuploidy populations, okay, they might somehow create an advantageous niche and then become resistance. So, and since we already know that aneuploidy cells has a lot of water influx, maybe this is something what water do, okay? We don't know what happened. And it might generate something more than just a turgor pressure. And especially that when you have a surface change, how does the host, okay, see these fungal pathogen? And then how these cells really survive from the adaptation, okay, during the adaptive process. So we don't know that, but our finding, we feel like now we open a big window to think about this question again, and beyond the genetics mutations or anything we think of before. So with that, I want to say that this morning I was making this slide, and I feel like I'm extremely lucky to have so many people supporting me along the way. And so the project was majorly supported by Ron in the Ronnie lab, and so we still keep working together. And Ben Lamon is a very talented. I feel like he's a genius in bioengineer. We are developing some interesting tools to detect antifungal resistance. Resistant cells, and we also do some work with Arturo Casadevil, okay, to look at the host pathogen interactions in the context of antifungal resistance. And last year, I joined the Happy Lab host and the pathogen interaction lab, and with Robin May, Liz Bloom, and Rebecca Drummond's lab. And starting a new lab in the pandemic is kind of weird. And without Liz Bloom, I don't think I can survive now. So, but our current work is supported by, we are really lucky, like supported by Royal Society and the University of Birmingham and the Institute of Global Innovations. And we have so many collaborators, and with my mentors and friends, and all have a very interesting collaborative projects going on now. So with that, I'm happy to take any questions later. So thank you.
All right, that was wonderful. Thank you so much. Thank you to both of our wonderful speakers today. So we're going to take some questions now. Let me try to get back. So, okay, so we have a question for our first speaker, for Elvis. The question from Jessica Brown is, it sounds as though there's a very low false positive rate for the lateral flow assay. So do you think that this is indicative of a low rate of latent infection, or does cerebral spinal fluid instead of, say, serum prevent false positives from latency?
Okay, thank you very much, Jessica, for the question. When I look at the question, I see two case scenarios. We have the latent infection in those who are asymptomatic, which is what we request for what we call systematic pre-antiretroviral screening. And then there is the search for cryptococcal antigen among patients with symptoms. And interestingly, when we look at all the studies which have been published so far on screening in advanced HIV, the positivity rate is around 6%. So, and given that the lateral flow assay has a very high sensitivity and very high specificity, we think that that is the prevalence among those who are asymptomatic. However, when we look among those who come with symptoms, the positivity rate is like 10 times that one. So in our paper, we found about 63%, which means that those with symptoms have a high propensity to have positive serum antigen. And that is one of the reasons why even among those who were asymptomatic and who were Crag positive, we tried to look at the data of those whom lumbar puncture was done. So even though just 6% of the patients who are symptomatic have a latent infection, the test is highly specific and highly sensitive enough to pick up these patients, and among them, 1 out of 3 have cryptococcal meningitis. So even among those who, the 63% who are positive with symptoms, close to 40% of them have cryptococcal meningitis. So another thing that we're also interested in is looking at the agreement between culture and cryptococcal antigen in cerebrospinal fluid, because we have to admit the fact that cerebrospinal fluid, to obtain cerebrospinal fluid is an invasive process. It needs some technical training. So we were trying to see how serum Crag, which is readily accessible, because there are studies which has been shown that even with finger prick, we can do serum Crag screening. How serum Crag can be used as a surrogate of the presence of cryptococcal meningitis especially in settings where we have really very rudimentary material. And as I mentioned, there is good news because even though there are relatively few patients with low CD4 who are Crag positive, and a high proportion with symptoms who are Crag positive, we are confident that the test is highly sensitive and highly specific to be able to pick up those who are positive and to be able to tell us that this one who is negative is actually negative. That is why, personally, I think that the development of the lateral flow assay, I mean, in the principle of the lateral flow assay, when evaluated and validated, was one of the best things that could happen in the diagnosis of cryptococcal meningitis. Yeah.
That's great. Thank you. Oh, yeah.
Yes, I wonder how you looked into other defining signatures behind crypto-histo and crypto-TB instead of only CD4. I think I like that question because my approach to patients is holistic, and one of the things which make clinical practice difficult is that we are very comfortable with conventions. The fact that the guidelines say you screen for TB, you screen for crypto. Once you've screened for those and you don't find them, but the patient is not fine, we're like, I don't know what this patient have, and we fold our arms. So my approach is to get people to start thinking, if not this, then what? And one of the most interesting areas is when you see patients with respiratory symptoms, they have hemoptysis, they have productive cough. You can also find that in histo patients. So if for once we think about TB, we don't find TB, and then we move forward to think about histo, and then maybe we say if we have access to bronchoscopy, we can send some fluid to look for pneumocystis jirovecii, then that's fine. So we looked at crypto in patients with advanced HIV. We are working hand in hand with those who are working on TB, but that doesn't mean we shouldn't bring in other fungal infections, because you will see most of the time we don't talk about pulmonary cryptococcosis in Africa. It looks like it doesn't exist. In Europe, we find them because clinicians are looking for them. There are diagnostic approaches, there are tools. So that is the type of holistic mind I really like young mycologists, and especially those who are interested in clinical mycology, to have that way of thinking.
That's great. Thank you so much.
Thank you.
We've got some questions in the chat for Hongji as well. I'm getting lost in the chat. So the first question from Herbert Debangi is as to whether aneuploid could be responsible for heteroresistance seen in azoles mainly.
I would say yes, but I don't know. We don't know, right? So there are some. I think there are some aneuploidy reports on Candida glabrata regarding aneuploidy and the heteroresistance. But to me, I'm more curious about how they got there. So, but this is a blank question. It's not as straightforward to say, but yeah, from our perspective in this talk, we don't know. Yeah.
Cool. And a question from Mark Voorhees, who asks, are Saccharomyces aneuploids more or less prone to filament?
We actually see a lot of filament-like shape in Saccharomyces cerevisiae when they become aneuploidy. This is very interesting because that there are some evidence in like mammalian cells that the osmotic stress, when you have a certain hypo-osmotic stress happen, the cell can move. And we also see that if there is, if those cells in a really confined environment, okay, their morphology will getting changed in like a microfluid chamber. So we see a lot of filamentation like structure in cerevisiae when they become aneuploidy. But is that the cause from an osmotic stress? We don't know. So
okay, very
we call those Pinocchio cells.
Pinocchio cells.
Yeah.
That's great. Oops. And another question. So how does the osmotic sensitivity of aneuploids affect survival of gut commensals like Candida?
This is a very interesting question. And we are actually working on some commensal strains now. And to be honest, I don't know, because we didn't really test the osmotic sensitivity. So I can be just honest saying, like, at this stage, we only look at some transcriptional response. Yeah. Okay. And to see if we can find something in common. Yeah, but we haven't, I really hope I can get into a lab to do those things.
Soon enough, hopefully.
Yeah.
When you say you're working with commensal strains, what does that mean exactly? You've isolated these sort of from like you've just isolated these from healthy individuals. Is that what makes them commensal strains versus others?
We get that from some animal models. Yeah.
Okay. Cool. And final so far question for you, Hongji, also from Mark Voorhees, is should other large scale changes in gene number, so for instance, expansion of LTR transposons in pathogens, could this lead to similar changes in turgor pressure?
So in our theory, this kind of turgor pressure change, osmotic balance, is triggered by the organic osmolytes and those small proteins, even amino acid or something like that. So, but regarding the LTR transposons, it really depends on where do they target, right? And also if there is an expansion, but those are non-coding. Those genetic element is not coding. So I don't think they will have a big osmotic change. Yeah.
Yeah. And Elvis, we've got a follow-up from your last statement from Jessica. So to follow up on your call for action, for those of us who are interested in drug or diagnostic development, what are important attributes for deployment in the clinic, particularly in resource-limited settings? So things like oral availability or side effects, et cetera.
Yeah, thank you, Rebecca, for the question. I think it's a very interesting question because when we look at the burden of HIV as well as its associated infection, the burden really lies in those remote rural areas. So as concerns diagnostics, most of the time it's not all the time that you have the opportunity of touching the patient. I'll use that in quotes, because some of the patients, they live like very far from the clinics, and they need to trek over long distances to get there. So when you have the opportunity, we try as much as possible to use the one-stop shop approach. It means that as concerns diagnostic approach, we would very much appreciate point of care tests. Tests that do not need so much technology to operate. Some of them should not need energy. And if they need energy, they should be able to use solar energy, for example. And in most of those clinics, we don't find like really qualified people. We may have a nursing assistant who is in charge of a health facility, in charge of about 1,000 HIV patients. So if these diagnostics are so easy to use, like the lateral flow assay, anybody can use it. You don't need to be a clinical personnel to be able to use it. And then the next thing is oral availability of drugs is really very important. And one of the things, if you look at, for example, the administration of flucytosine has to be in 4 divided doses. And even if you're doing direct observation of the treatment, it's not really very feasible. So as concerns their drugs, oral availability, facility to store the medications, as well as long-acting medications. Just like we're talking about long-acting medications for HIV. So for those who are really interested in the development of diagnostic tests and treatment, I agree that you can only know about the side effects when there has been a massive rollout of the treatment and monitoring. But if they can focus on producing slow release tablets, the one that you can take for once a day and it covers 24 hours, if the point of care test, if the test can be point of care with very easy to use as well as easy to store, then that would be the best approach. Even in the clinics, in the very advanced hospitals, you know, at times bureaucratics may make the diagnosis to take a very long time. If you have a point of care test while you're waiting for the lab guy to put on his very sophisticated machines, you already know what the patient has, and you already institute your treatment. So I think that's the way forward.
That's really interesting. And do you know if there are any sort of current existence of tests like that, or that are currently under development in any stages? Like, is this something that is making its way into clinical practice?
Okay, I think as concerns the tests, so far there was discussion about a lateral flow assay type of CD4 test, which classifies the patient either greater than or less than 200 CD4, because for those of us who are interested in advanced HIV, we are still complaining about the fact that CD4 was taken away from the follow-up of patients. And now we are trying to say, even if you don't want us to do CD4 as a monitoring tool for patients on antiretroviral therapy, make it available for us when we suspect advanced HIV. And that's one of the reasons why I said any unwell patient with HIV who comes to the hospital, even if he's complaining that he has, I don't know, he had a scratch by his cat, it's an opportunity to screen them for advanced HIV. So one of the one we've been hearing about is true. I haven't really looked into it in detail. It's the one where you have like a lateral flow assay to classify the patient either lower or greater than 200 CD4. And then we were also advocating for the production of slow-acting flu cytosine. With the COVID-19 and all the trouble which is going on now, it's very difficult to know where they are with that.
Right, absolutely. Okay, very interesting. Thank you. We've got another question for Hung Ji. So great talk, Hung Ji, says Lillian. Looking at your proteome data, do you see any correlation between pathways involved in stress responses and the drug-resistant aneuploid strains? So would you expect pathways normally associated with stress responses to be constitutively on in an aneuploid? Cool question.
So yeah, this is a great question. Thank you, Lillian. So I want to clarify a little bit about this. So the reason, so we do the stress response correlation at the beginning when we look at the aneuploidy transcriptional signature, is because we are analyzing a population, okay? And this is a very mixed population, and we actually don't know what we are looking at if we look at a single pathways. So that's why we take the entire genome genomic expression pattern to compare with other stress response pattern to think about this question rather than dissect what's the pathway. So from the data I have currently, we cannot tell anything. And I'm also worried it would be misleading if we do that. So we never defined it. So, but we do have the plan to really look at that in a more systematic way in Candida albicans. So, so now we are trying to develop new methods to purify Candida albicans aneuploides, okay, without too much selection force. Yeah, so this is ongoing things to do. But actually, I was only thinking about how the stress response was altered, but your question actually is better. It's a better idea. Thank you.
Very cool idea.
Yeah.
All right, we don't. Oh, sorry. Go ahead, Herbert.
No, I was just making. No, nothing much. Nothing much.
Okay.
Go
Sorry.
on.
So I don't think we have any other questions unless there are any other questions remaining. Stimulated